This video will go through subtopic 3.1, genes. Here are the list of understandings, application, and skills that will be covered in this video, as well as some of the things that you should be able to know, as well as things that you don't really need to know. So we'll start with a quick overview of a few different units that were covered previously in the course, looking at some of the information that you'll need to know going forward into the genetics unit. So just reminders that with hereditary information, that hereditary information is contained within DNA. All those traits and all those proteins are encoded within that DNA found within the nucleus of the cell. That DNA isn't used directly to make a protein, but is instead used to create a message molecule, that RNA. That RNA is then used to create that protein. And that protein can have various functions, so it can be structural or it can act as an enzyme. If it acts as an enzyme, it's going to do different functions, and this can ultimately control traits that are expressed in an organism. For example, things like eye color or hair color. And so within the genetics unit, we'll talk about how DNA is used to control these traits through these specific segments of DNA referred to as a gene, the specific parts of DNA that will encode a protein. And just a reminder that with that code for making a polypeptide or for making a protein, that genetic code is considered to be universal. So the same three letters that would go into making up any amino acid or encoding an amino acid in RNA is going to be the same three letters across different species and across different organisms, and will be similar across prokaryotes and eukaryotes. So it really is kind of a universal code. So a little bit more background information about DNA before we get going. So DNA is that hereditary information containing all that information for those traits. DNA has to be duplicated, so an exact copy is passed to daughter cells, and the number of cells are doubled, but DNA stays the same, so the quantity of DNA within a cell does not increase, even though the number of cells will increase. Transmission of hereditary material is referred to as genetic continuity, and long strands of DNA are packaged in condensed folded structures to make chromosomes, and it's in the chromosome form of DNA that we will refer to this hereditary information as. And just a reminder that chromosomes are made up of DNA and proteins, those proteins being histones. So what is a gene? A gene is basically a heritable factor that consists of a length of DNA and influences a specific characteristic. Or a gene is a small length of DNA that will encode a protein, and that protein will control that specific characteristic. Eye color, hair color, skin color, whatever else. Now, within humans, there are 46 chromosomes within a cell, and on those 46 chromosomes, they contain thousands of genes. So genes are located on chromosomes, which would make sense considering that a chromosome is a folded DNA structure. So each gene consists of a much shorter length of DNA than in chromosomes, so many genes are found on that much longer length of chromosome. And so we can then say that each chromosome carries many genes. So when comparing the number of genes across species, Ivy makes a specific point of saying that the number of genes should not be referred to as genome size. So genome size is a specific term that's used for the total amount of DNA within a cell. So that total amount of DNA includes segments of DNA that do not include or encode for genes, as well as mitochondrial DNA if it's an animal cell, And if it's a plant cell, it would include the DNA within the nucleus plus mitochondrial DNA and then DNA within the chloroplast. And so the number of genes within different organisms will vary greatly. So here we have like bacteria, relatively small number of genes. You can get into things like protists here that actually have more genes than humans. So here's a human. We have about 23,000 genes encoded within our 46,000 chromosomes. And then, you have something like rice that has 41,000 genes, so way more genes than humans have. So the number of genes will vary between species. So with these genes in different species, similar genes can be found in different species. And these genes may differ by a few DNA nucleotides. So those letters that encode DNA, the A, the T, the C, and the G, those similar genes found in different species will only differ by a few of those letters. And these differences in these letters can cause, for example, a change in the base sequence of the gene. Remember, you're now changing some of the letters, which can change, which will ultimately change the sequence, the base sequence or the sequence of letters. And because we're changing the sequence of the letters in the DNA, this can cause a change in the mRNA made in that transcript. And because we're changing the letters in the mRNA, and remember those specific codons will encode for a specific amino acid, this could potentially cause a change in the amino acid sequence and ultimately change the primary structure of the protein, that sequence of amino acids. That change in the primary structure can affect the secondary, tertiary, or quaternary structure of the protein. So ultimately how that protein folds and how that protein interacts with other subunits. Ultimately, if we change the shape, if we change the tertiary structure of the protein, this will cause a change in the shape. And if we change the shape, this could change the function of the protein. So there could be slight variations in how these proteins, encoded by similar genes, function across different species. One example that we'll discuss in class is a gene that's associated with language in humans. So where are genes located? Well, genes will occupy a specific position on one type of chromosome, meaning that, within humans, for example, a gene for hair color will always be located on the same chromosome and will be located on the same position on that particular chromosome. This position is referred to as the locus of the gene. Now, genes can also have different forms. So if we continue with our example of hair color, you know that many different types of hair color. So that would suggest that there are different combinations of genes or different types of genes or variations of those genes that would control hair color. And so in genetics, we refer to various forms of a gene as an allele. So, you change the sequence a little bit, but encode for the same protein within a species, you get what is called an allele. Alleles will occupy the same locus, the same position of the chromosome, but will have different variations of the gene. So most plant and animal cells have 2 copies of each chromosome, so we can expect 2 copies of a gene to be present. So every cell will have 2 copies of the same chromosome. One would be from mom, one would be from dad, so we expect 2 copies of each gene to be present. And so these could be 2 of the same allele, or they could be different. So what causes these differences between these genes to give rise to alleles? So alleles will differ from each other by 1 or a few nucleotides. So for example, one allele might have an adenine, have an A, and the other might have a cytosine, that's C, at the same position. positions in a gene where more than one base may be present are referred to as SNPs, single nucleotide polymorphisms. So a lot of words there, so just a single nucleotide where many can be, where you can have different nucleotides present. So poly meaning many, morphisms meaning changes. So many changes in that single nucleotide area. So the following diagram shows an example of that SNP. So for example, you'll notice that this gene sequence, this base sequence, is the same for all three of these genes. But in this one position, you can have variation. Instead of that A, you could have a G, or you could have a T. And that substitution, that variation in this one position can give rise to different forms of that gene, so different alleles. Now, several SNPs may be present in a gene, but even then the alleles of the gene may only differ by a few bases. So it's not going to be dramatic differences between these alleles. Ultimately, you can't change the sequence of the gene too much without ultimately affecting what that protein will do, or how that enzyme or protein will behave. So ultimately, SNPs are only going to be present in a few bases within a gene, at most. Now, how do these SNPs arrive? SNPs, or these single nucleotide polymorphisms, can arise by mutation. So mutations, at its most basic, are going to be considered spelling mistakes in the DNA sequence. And basically, as DNA gets copied, different bases could get accidentally inserted by the DNA polymerase, and so it results in a spelling mistake, essentially. So new alleles basically are formed by mutation of other alleles or genes that exist. Most mutations are neutral, meaning there is no noticeable effect, or harmful, but occasionally they can be beneficial. Now, mutations may cause a change in the mRNA that's made in transcription, a change in the amino acid sequence, which will then cause a change in the secondary and tertiary structure, or even the quaternary structure. A change in that tertiary structure is going to cause a change in the shape of the protein, and if we change the shape of the protein, you may get a different function of the protein, or you may alter the function of the protein slightly. Now, there are two main types of mutations. There is what's called point mutations. This is where a single nucleotide base change of the DNA results. So point mutations can take different forms. So you can have what's called a silent mutation, nonsense mutation, or a missense mutation. Now, here we have no mutation, so this is considered like the wild type version of the gene, or the original version of the gene. So here we have the original DNA sequence. Here we have what's coded for at the RNA level, and then this codon giving the amino acid that it would code for at the protein level. So with a silent mutation now, like a change in the last letter to a T, so from a C to a T, ultimately does not change the amino acid that is coded for. So it doesn't change anything about the protein, which is why it's called a silent mutation. You don't notice that a mutation has happened. Now there's what's called a nonsense mutation. So in this case, if you change the first letter of the base at the DNA level, you completely change what the codon codes for, and it will code for basically a stop codon, and you'll get premature termination of protein coding. And then you can get missense mutations, which are conservative or non-conservative. So if you change the middle letter, you can get a different amino acid being encoded for, and it's considered conservative because you're basically replacing one charged amino acid with another similarly charged amino acid. So while they may be different, they should behave the same and not affect protein function too much. then you can get non-conservative mutations, which give rise to a completely different type of amino acid that has different functional properties. Now, there's what's called frame shift mutations. This is basically the addition or deletion of a base pair. So here, if we have our wild type gene, so our DNA sequence before mutation, if you have a random insertion while DNA is being copied of a base, so here we have the ACA sequence, and then when that DNA gets copied, a G gets randomly inserted. And so this completely shifts the codon sequence now. So here we have codon 1, which gets preserved, but now codon 2 is something completely different. So codon 2 that would originally code for a lysine now codes for glutamic acid, which completely shifts how all the other codons are read. Now, with a deletion, this is your DNA copying, a base gets removed or gets left out for whatever reasons. So again, here we have our first codon coding for threonine. Then we have that second A that gets removed, and again completely shifts the reading frame, which is why it's called a frame shift mutation. So now the codon sequence is completely changed again. So this diagram here just illustrates like the different effects graphically of different types of mutations. So if we have the normal, and then here is our sequence of letters, you know, basically like if you start to change the letters around, you get different interpretations or different results. So with the substitution, you know, you're swapping one letter for another. So in this case, beast becomes feast, and it means something else. If we insert a letter, for example, an S at the end, we get beast, and you know, we get double the beast now. With a deletion, you know, we're removing a letter, so we're taking out an A, and now instead of beast, we get best. And if we invert our sequence of letters, in this case the T and the S, instead of beast, we now get beats, and you know, we can enjoy some music. So ultimately, these mutations can give rise to variations in genes that can have differing effects and can give rise to genetic diseases. One common genetic disease is sickle cell anemia. So sickle cell anemia is a genetic disease and the result of a point mutation in the gene that encodes for alpha globin polypeptide, or basically the alpha globin of hemoglobin. And the short form of the gene is encoded with this capital H, lowercase b, so this basically means hemoglobin. And so most humans have what is referred to as the HbA, and we use this superscript A to indicate the normal version or the wild type version of the gene. So sickle cell disease occurs when a person inherits from mom and from dad two abnormal copies of a hemoglobin gene. A person with a single abnormal copy does not experience symptoms, and it has some protection from the disease malaria, but is able to have normal functioning red blood cells. And basically, you're only going to inherit a mutation if the mutation occurs in gamete cells, and remember those gamete cells live in the ovaries and testes. So with sickle cell anemia, how this arises is basically through that base substitution that will convert the 6 codon of the gene for hemoglobin from GAG to GTC. And basically you get a new allele that is formed that is referred to as the sickle cell allele. So this is abbreviated as HB superscript S. So transcription of that 6th amino acid now will result in a valine instead of a glutamic acid. And so basically this glutamic acid is a polar amino acid that is now getting replaced with a non-polar amino acid. So this change will cause hemoglobin molecules to stick together because of that exposed valine being non-polar, not wanting to interact with the polar surroundings, will cause these hemoglobin molecules to stick together, and especially stick together in tissues with low oxygen concentration. The bundles of hemoglobin molecules formed are rigid enough to distort the red blood cells into a sickle shape. So instead of having a nice round shape, they basically have this sickle shape to them, or this crescent moon shape to them, hence the name sickle cell anemia. And so this diagram here illustrates the mutation that occurs from the normal DNA sequence to the sickle cell mutation, and how that affects the amino acid that is encoded in the protein. So you normally get that glutamic acid, and instead you get valine encoded in that sickle cell gene. So with sickle cell anemia, these sickle cells will cause damage to tissues by becoming trapped in the capillaries, blocking them and reducing flow. When sickle cells return to high oxygen levels in the lung, the hemoglobin bundles will break up and the cells will return to their normal shape. So basically, like in the capillaries, you get the creation, or these cells folding into this sickle cell shape, and then returning back to this normal round shape in areas of high oxygen concentration. So these cells go back and forth between these shapes as they travel within the body. So both the hemoglobin and the plasma membrane are damaged as a result of this, and the life of the red blood cell is reduced. So a typical lifespan of a red blood cell is about 120 days, and that gets greatly reduced. So the body cannot replace red blood cells at a rapid enough rate, and anemia develops. So kind of broadly looking at some of the terms in genetics, is this idea of a genome. So what is a genome? So a genome is the entire base sequence of each DNA molecule. So in humans, for example, there are 46 molecules that form the chromosomes in the nucleus. So we have 46 chromosomes. And our genome would include the DNA within our mitochondria. So a genome is the entire sequence of all the DNA within the cells of our body. In plants, as I mentioned before, the genome would include the DNA molecules of the chromosomes, plus the DNA of the mitochondria and the chloroplasts. And in prokaryotes, it would include the circular chromosome, plus any plasmids as well. So, in the early part of this century, and in the latter part of the last century, there was a great endeavor called the Human Genome Project, which looked to sequence the entire genome. And this was a project that began in 1990 and was completed in 2003. So it took 13 years to sequence a human genome. So the project drove rapid improvements in base sequencing techniques. When it first started, the techniques for sequencing the letters in a genome, wasn't very good. Took a lot of labor and a lot of effort, but towards the end of it, how quickly they could sequence those letters rapidly improved. So the Human Genome Project, while it did give us a sequence of a human genome, did not give us an immediate and total understanding of human genetics. It just kind of really allowed things to kind of get going, and really genetics of the human genome to be studied. What it did tell us, though, is what genes are protein coded. And it told us that there were about 23,000 protein coding genes, a lot less than what's expected, considering that you have about 100,000 proteins within the human body. Most of the DNA is not transcribed and is generally referred to as junk DNA, but we now know that it is not actually junk DNA and will actually help with transcription of protein coding genes. They also found a lot of satellite DNA, which is considered highly repetitive DNA, found towards the centromeres and the telomeres, which are the ends of the chromosome. And basically, that Human Genome Project was a race to sequence. So there were two parts, or two competing projects for the Human Genome Project. There was the public project, which was government funded and carried out in academic institutions. And then there was a private company called Celera, founded by a scientist named Craig Venter. Ventnor planned to sequence the Human Genome Project within 3 years, and this was going to be well ahead of the public effort. So Celera actually brought competition between the private and public project and pushed the public effort to kind of step up its game. Now, the public project did have quite a bit of a head start, but that head start was not strong. And considering the technology that Ventnor was bringing in and developing, he was able to quickly catch up to that public project. Now, in February of 2001, both groups separately published draft sequences of the Human Genome Project in Science and Nature, and they agreed to publish them at the same time. So there were ultimately two methods that were used to sequence the human genome. You do not need to know the details of this, but these two methods are what's referred to as the hierarchical shotgun sequencing method and the whole genome shotgun sequencing method. So the details of how this was done, you don't need to know or worry about. Now, the Human Genome Project, it's important to note that it sequenced one set of chromosomes. So it basically sequenced a human genome, but not the human genome. When you consider all the different alleles that exist within the human population, sequencing one set of chromosomes is not sequencing the genome for the human species. So the Human Genome Project is still looking for variations in that sequence, and most DNA is shared between humans, but there are SNPs, those single nucleotide polymorphisms, which contribute to diversity, and it's generally those that are of the most interest. And since publication of the human genome, the base sequence of many other species has been determined. So this technology has been used to sequence the genome of many different species. And this allows us to compare these genomes. And when we compare these genomes, they show aspects of evolutionary history of living organisms. And we'll get into this in the evolution unit as one aspect of evidence for evolution. So what we'll talk about in class is how sequencing is done, and particularly how sequencing is done using a method called the Sanger dideoxy DNA sequence. So that is the end of this video for 3.1.